In this video, we will be discussing about bacterial structure. Since it's quite a long topic, I'll cover it in parts. In part 1, I will be covering the concepts of prokaryotes and eukaryotes, introduction about bacterial structure, and details of bacterial cell wall. In the end of the video, I'll share with you the summary of the concepts covered today as well as a few MCQs. Let's begin with the 5 kingdom classification. According to this classification, living organisms can be classified into 5 kingdoms that include Plantae, fungi, Animalia, Protista, and Monera. Bacteria belong to the class Monera. These organisms are prokaryotic as well as unicellular in nature, and some of them have cell wall too. Now let's understand what does prokaryotes and eukaryotes mean. It's a major biological division of organisms in which eukaryotes includes organisms who have membrane-bound nucleus. On the other hand, prokaryotes have a genetic material that is not physically separated from cytoplasm. Eukaryotes are larger in size as compared to prokaryotes. Eukaryotes include algae, protozoa, fungi, and slime molds. On the other hand, prokaryotes include bacteria. Eukaryotes and prokaryotes are organisms since they have all the enzymes and organelles that are necessary to replicate and generate metabolic energy. This differs them from viruses which depend on host cell for replication and generating their metabolic energy. Now coming to the differences between prokaryotes and eukaryotes. In this table, we can see the major differences at a glance. When we look at the nucleus, the nuclear membrane, nucleolus, DNA, and mitosis, these 4 factors are absent from prokaryotes but they are present in eukaryotes. When we look at the chromosome, we see that prokaryotes have a single circular chromosome whereas eukaryotes have more than one chromosome and that too linear in nature. Now coming to the cytoplasm. Cytoplasmic streaming, pinocytosis, mitochondria, lysosome, Golgi apparatus as well as endoplasmic reticulum. All these are absent from prokaryotes, but they are present in eukaryotes. We'll go into the detail of each of these structures later on. Coming to the cell wall. Sterols is present in eukaryotes, not in prokaryotes, and muramic acid, which is a typical structure of prokaryotic cell wall, is absent from eukaryotic cell wall. Now coming to the prokaryotic cell structure. Prokaryotic cell is simpler as compared to eukaryotic cell, except the cell envelope. There is a concept of nucleoid in prokaryotes in which the chromosomal DNA is folded and packed in a specialized region. This specialized region is known as nucleoid. Nucleoid of most bacteria consists of a single circular chromosome. But there are a few exceptions. Like few bacteria have 2 to 4 dissimilar molecules. We can see this in case of Vibrio cholera and Brucella melitensis. These two organisms have 2 dissimilar chromosomes. Also, Borrelia burgdorferi and Streptomyces silicolor, they have linear chromosomes instead of circular chromosomes. Prokaryotic cell structure. When we look under a microscope, we can see that in prokaryotic cell there is no nuclear membrane. Again, there is an exception, that is planktomycetes. In this organism, which is an aquatic bacteria, nucleoid is surrounded by nuclear envelope. This is against the usual law of prokaryotic cells. Moreover, in prokaryotic cells, we have seen no mitotic apparatus. So among these two differences, this no mitotic apparatus still holds true. Number of nucleoids or chromosomes depends on the growth condition, that is, they are more in more rapidly growing organisms. However, prokaryotic cells are haploid by nature. Now let's discuss the morphology of bacteria. Bacteria are usually 0.2 to 2 micron in diameter and 1 to 6 micron in length. This is a diagram showing a basic structure of bacteria. The outermost layer, which is pink colored, is the capsule. Within this there is a purple colored thick layer which is cell wall, and inside it is a thin layer of plasma membrane. Between the plasma membrane and cell wall we can see some space. This is periplasmic space. Moreover, plasma membrane is involuted at a place to form mesosome. Now within this cell envelope, which includes the cell wall and membrane, there is cytoplasm and there are various structures in cytoplasm. In the center we can see there is a single circular chromosome which is typical of bacterial cell. Moreover, there is an additional genetic material called plasmid. There are few cytoplasmic structures like cytoplasmic inclusions and ribosomes. Ribosomes are the place where protein synthesis takes place. Then there are additional appendages, that is flagella. Flagella helps in the movement of bacteria, and pili. Coming to the coming to the chemical composition of cell wall. Cell wall is basically made up of peptidoglycan. This peptidoglycan is also known as mucopeptide or murine. So as the name suggests, peptidoglycan means it is made up of two parts, that is peptide and polysaccharide. The cell wall of gram-positive organisms is much simpler when we compare it with that of gram-negative organisms. And what is gram-negative? Bacteria are classified as being gram-positive or gram-negative based on the color they take on gram stain. This gram stain was introduced by a histologist whose name was Hans Christian Gram in 1884. It's a type of differential staining technique in which bacterial smear is first subjected to a primary stain. This primary stain can be crystal violet or methyl violet or a gentian violet. This is followed by a decolorizer. Decolorizer can be ethanol or acetone or aniline, and then they are subjected to a counter stain which can be either safranin, carbon fusion or neutral lead. So these two primary stain, this is basically crystal violet is purple colored, and the counter stain safranin, this is pink color. So gram staining depends on the ability of some bacteria, that is the gram positive bacteria, to retain the diiodine complex after decolorization. Whereas in case of gram-negative bacteria, they do not retain diiodine complex and they get decolorized on treating with decolorizer. Hence, they take the counter stain, that is this one, second one stain. Thus, gram-positive bacteria appear purple, whereas gram-negative bacteria appear pink after gram staining. So, this difference is attributed to the difference in their cell envelope. What is the principle of gram staining? Gram staining basically depends on the different in composition of the cell wall of these two organisms. Gram positive bacteria have more acidic cytoplasm and hence they retain the diiodine, that is the primary dye, crystal violet and iodine complex pattern as compared to the gram negative. Now we have seen that peptidoglycan layer. Peptidoglycan layer of gram positive is thicker and hence they retain the diiodine complex pattern. On the other hand, gram negative organisms, they have more of lipid content which makes them more permeable to the secondary dye after it has been treated with a decolorizer. So they take a secondary dye because we have treated it with an organic solvent which dissolves the lipid part of the gram negative organism. Now let's discuss the components of gram-positive and gram-negative cell wall. Cell wall of gram-positive organism consists of peptidoglycan, teichoic acid and polysaccharide. On the other hand, in case of gram-negative organism, the cell wall consists of peptidoglycan, an outer membrane, lipopolysaccharide and lipoprotein. Now let's have a look at the structure of cell wall of gram-positive and gram-negative organisms. On the left side, there is a structure of gram-positive cell wall. This you can clearly see is simpler when we compare it with the cell wall of gram-negative organism. So after a plasma membrane, this is not a part of cell wall, there is a layer of peptidoglycan which is thicker as compared to that of gram-negative organisms. And you can see here is a space between peptidoglycan and the cytoplasmic membrane. This space in gram-positive is called as the periplasmic space or periplasm. If the bacteria has a capsule, they will be having capsular polysaccharide and capsular protein on the surface as well. So this is a basic simple structure of a cell wall of gram-positive organism. Now let's have a look on the cell wall of gram-negative bacteria. So after plasma membrane, there is a peptidoglycan layer which is thinner as compared to that of the gram-positive organisms, and we have already discussed this. This is followed by an outer membrane. This outer membrane is a bilayered structure. It has an inner leaflet. This is the inner leaflet which mimics plasma membrane. The properties of this inner leaflet is that like that of the plasma membrane, and it also has an outer leaflet which has a spatial component. This spatial component is called as lipopolysaccharide. So the two leaflets are asymmetrical and their properties also differ from that of the plasma membrane. Outer membrane also has a transmembrane protein. This is a channel protein which is called as porin. This porin allows diffusion, that is passive diffusion of low molecular weight hydrophilic compounds through it. When there are larger compounds, larger molecules like antibiotics, they find it difficult to penetrate. Hence, there is more resistance of antibiotics in case of gram-negative organisms. At the inner leaflet, you can see there is a lipoprotein. which connects the outer membrane with the peptidoglycan layer. On the surface, there is lipopolysaccharide, and this lipopolysaccharide is a complex molecule. It consists of lipid A plus a polysaccharide core and O antigen. Here the space between outer membrane and inner membrane, this is called as a periplasmic space. So this is the basic structure of cell wall of gram-negative organisms. Now coming to the components of cell wall. Cell wall has a peptidoglycan layer, we have already discussed. So what is this peptidoglycan? It is actually a complex polymer and consists of three parts. It has a backbone of N-acetyl glucosamine or NAG. It is being depicted in a red circle here. And N-acetyl muramic acid, which is depicted by a blue circle here. So this will form the backbone of peptidoglycan, N-acetyl glucosamine and N-acetyl muramic acid. Further, if we see, then there are tetrapeptide side chains. This is tetrapeptide side chain, and the two layers are cross bridged with the help of a pentapeptide cross bridge. This cross bridging provides rigidity to the cell wall. If we see, then here the N-acetyl glucosamine and N-acetyl muramic acid, they are same in all species, but these tetrapeptide side chains and pentapeptide cross bridges, they may vary between different species. Peptidoglycan layer is a single large molecule. In case of gram-positives, there are about 40 sheets of peptidoglycan, which consist of up to 50% of the cell wall. Whereas in gram-negative, as we have already seen, it is thinner, there are only 1 to 2 sheets of peptidoglycan, and it consists of only 5 to 10% of the cell wall. So there is an important question which covers that the thickness of gram-positive of peptidoglycan in gram-positive organisms is 15 to 80 nanometer, whereas in case of gram-negative it is only 2 to 3 nanometer. In tetrapeptide side chain, position 3 is most variable one. Most gram-negative organisms have a special component called diaminopymalic acid at this position. So earlier we have seen there was a tetrapeptide chain, that means there were 4 peptides. So among that, at position 3, most gram-negative organisms have this component which is called diaminopymalic acid, and this is a unique component of bacterial cell wall, as it is never seen in case of archaea or eukaryotes. Now let's have a look at gram-positive cell wall. Apart from the peptidoglycan, what they have? They have a teichoic acid and polysaccharide. Teichoic acid provide negative charge to the cell surface, and they are of two types, that is a wall teichoic acid and a membrane teichoic acid. Wall teichoic acid is attached to the peptidoglycan layer, whereas membrane teichoic acid goes all the way to the plasma membrane. Wall teichoic acid is covalently linked to the peptidoglycan, as we have seen in the earlier diagram, whereas membrane teichoic acid, it is covalently linked to the membrane glycolipid, hence it is called as lipoteichoic acid. So membrane teichoic acid is known as lipoteichoic acid. This is an important component. Composition of teichoic acid vary with the growth medium in which the bacteria is grown. It will vary. They are major surface antigens of gram-positive organisms containing ribitol or glycerol polymers. For example, if we see Streptococcus pneumoniae, they have teichoic acid which act as antigenic determinant, and this antigen is called as Fosman antigen. Now the polysaccharide component. Polysaccharide component includes mannose, arabinose, rhamnose, glucosamine, glucuronic acid and mannuronic acid. Coming to the gram-negative cell wall, as we have already seen, there was an outer membrane, lipoprotein and lipopolysaccharide. Outer membrane was bilayered with an inner leaflet and an outer leaflet. Inner leaflet was similar to cell membrane and outer leaflet had lipopolysaccharide attached to it. Now let's discuss about outer membrane proteins. We have seen there was a porin. Porin are special transmembrane channel proteins for passive diffusion of hydrophilic compounds, and they also act as receptor for some bacteriophages. Large antibiotics penetrate slowly, hence there is a high degree of resistance in gram-negative organisms when we compare it to the gram-positive organisms. Several other proteins are also present, which are involved in the transport of molecules or enzymes like proteases, phospholipase. Outer membrane is connected to peptidoglycan layer by outer membrane lipoprotein. This also we have seen. It is also connected to the cytoplasmic membrane. Now let's come to the lipopolysaccharide part. Lipopolysaccharide consists of three regions, which includes a glycolipid. It is called as lipid A and it is attached to a polysaccharide. This polysaccharide consists of a polysaccharide core as well as terminal repeat units called O antigen. This O antigen is responsible for the O antigen specificity of the bacteria. Lipopolysaccharide is synthesized on cytoplasmic membrane and then it is transported to the exterior. Lipopolysaccharide is negatively charged and cross-bridged by cations like calcium ions, magnesium ions. This stabilizes the membrane and is a barrier to hydrophobic molecules. Lipopolysaccharide in gram-negative is also called as endotoxin. This is an important component, is strongly bound to the cell surface and is released only when the cell is light. That's why endotoxin. Endo means it is bound to the cell surface and it will be released only when the cell will lyse. This lipopolysaccharide is responsible for endotoxicity and O antigen specificity, as there was specific repeated units O antigen which provides O antigen specificity to the organism and it also provides endotoxicity. Toxicity is mainly because of the lipid A part. We have seen that there was a lipid A part, there was a polysaccharide core and there was O antigen. So this endotoxicity was because of lipid A part. What does endotoxicity includes? It includes pyrogenicity. Pyrogenicity means induction of fever. Lethality means ability to kill. Tissue necrosis means damage to the tissue. Anti-complementary, it will act against the complements of the body. And anti-tumor activity, that is, it will kill the tumor cells. Now coming to the lipoprotein. Lipoprotein cross-links outer membrane with the peptidoglycan layer. And its main role is to stabilize the outer membrane. Now let's come to the periplasmic space. What is this periplasmic space? As we have already seen, it is the space between the outer and the inner membrane in case of gram-negative organisms. And in gram-positive, it is the space between cytoplasmic membrane and the peptidoglycan layer. So in gram-negative, it includes a peptidoglycan layer and a gel-like component of proteins. These proteins are called as periplasmic proteins. These periplasmic proteins will include binding proteins, which are the binding proteins for sugars, amino acids, and hydrolytic enzymes. These hydrolytic enzymes like alkaline phosphatase. They can be detoxifying enzymes like beta lactamases. Beta lactamases are actually the enzymes which damage a lot of antibiotics like penicillins, cephalosporins, carbapenems. Then finally there are membrane derived oligosaccharides. These help in osmoregulation. So basically the periplasmic space will occupy about 20 to 40% of the cell volume. So this is a basic table showing difference between the cell wall of gram-positive and gram-negative organisms. As we have already seen, the cell wall of gram-positive is thicker, that of the gram-negative is thinner. Variety of amino acids, there is fewer variety of amino acids in gram-positive, but there are several amino acids in case of gram-negative organisms. Lipids also, they are absent in gram-positive, but they are present in gram-negative. Teichoic acid we have seen is a component of gram-positive cell wall, but it is not there in the gram-negative cell wall. Most important is the thickness of peptidoglycan, which is about 15 to 80 nanometers in case of gram-positive, and it is only 2 to 3 nanometers in case of gram-negative organism. Now what is the function of cell wall? Function of cell wall is to maintain the shape, size and rigidity of the cell. It provides strength, ductility and protection against osmotic damage. So if the bacterial cell wall is gone, it will lyse due to osmotic damage. It also takes part in cell division. It is a target site for various antibiotics like beta-lactams, lysozymes and bacteriophages. It carries bacterial antigen. As we have seen, there was a Fosman antigen present in Streptococcus pneumoniae. They have a role in virulence as well as immunity because antibodies will be produced against these antigens. Lipopolysaccharides in gram-negative bacteria have endotoxic property. We have also discussed this in detail. Now, how can we demonstrate the presence of cell wall? It can be demonstrated by various methods like by plasmolysis. When the bacteria is placed in a hypertonic saline, then water is lost from the cytoplasm by osmolysis and cell shrinks while the cell wall stays intact, and this is known as the bacterial ghost. There are various other methods like microdissection, or reacting the organism with antibodies, mechanically rupturing them, differential staining technique like that of the gram stain, electron microscopy. Now coming to the defective form of cell walls. What are they? They include protoplast, spheroplast or L forms. These are the cells without a cell wall. It can be formed due to exposure to hydrolytic enzymes like lysozymes or by blocking the peptidoglycan synthesis, which can be done with the help of beta-lactam antibiotics like penicillin, cephalosporins, carbapenems. This releases protoplast in case of gram-positive and spheroplast in case of gram-negative organism. If such cells are able to grow and they divide, then what they form? They form the L forms. These L forms are actually difficult to grow and hence they need a solid media like agar for their growth. They are produced more readily with penicillin as compared with lysozyme, and they can also revert back to normal if the stimulus is the lysozyme or penicillin. If they are removed, they can revert to their normal form also. But few of them are stable and cannot be reverted back. Some L forms are produced spontaneously in host may cause various chronic infections. Now coming to the mycoplasma. Mycoplasma are actually cell wall lacking bacteria and have no peptidoglycan layer. There is no target for antibiotics and hence they are resistant to the cell wall inhibiting antibiotics. They differ from L form. Why? Because in case of L form we have seen that they revert back to their original form once the stimuli is removed. But mycoplasmas do not revert back to a cell forming bacteria. It has also been seen that there are few archaea which are cell wall. Now coming to the summary of concepts we have covered so far, bacteria belong to the kingdom Monera. They are prokaryotes and hence they have no membrane bound nucleus and no mitotic apparatus. We have seen that they have a single circular chromosome and this chromosome is folded in a specialized region which is called as nucleoid. Bacterial cell is about 0.2 to 2 micron in diameter and it is about 1 to 6 micron in length. Gram stain is a differential staining technique which is based on the differences between cell wall of gram-positive and gram-negative organisms, and it helps to differentiate bacteria into being gram-positive or gram-negative. This is a diagram showing difference between gram-positive and gram-negative cell wall. In this area is the cytoplasm or the inside of the cell, and this is the outer part. So in the cytoplasm we see there is a cytoplasmic membrane just adjacent to it, followed by a space which is called as the periplasmic space, and then this blue structure is the peptidoglycan layer. In case of gram positive this peptidoglycan layer is thick, whereas in case of gram negative it is thin. Then looking at the gram negative organisms, inside the cytoplasm the adjacent structure is cytoplasmic membrane followed by a space and a thin layer of peptidoglycan layer. After that there is again some space and a layer of outer membrane. This part is outer membrane. In the outer membrane we have various proteins like porin which serves as a channel for diffusion of molecules. And then there is a lipoprotein. Lipoprotein attaches the outer membrane to the peptidoglycan layer. On the surface there is lipopolysaccharide. This lipopolysaccharide is made up of three components: lipid A, polysaccharide core and O antigen. This is responsible for endotoxicity and O antigen specificity of the organism. So periplasmic space is the space between outer membrane and cytoplasmic membrane in case of gram negative, whereas in case of gram positive it is the space between the peptidoglycan layer and cytoplasmic membrane. Now the main component of cell wall is peptidoglycan, which is also known as mucopeptide or murine. This peptidoglycan has a backbone of N-acetylglucosamine and N-acetylmuramic acid with the repeating units. These two units are linked by beta-1,4 glycosidic bonds, and then there is a tetrapeptide side chain attached to N-acetylmuramic acid and pentapeptide cross bridges which provides rigidity to the cell wall. So in case of gram positive organisms, other than peptidoglycan which is thick 15 to 80 nanometer in size, other components are teichoic acid and polysaccharide. This teichoic acid can be wall teichoic acid or membrane teichoic acid. Wall teichoic acid goes up to the peptidoglycan layer, whereas membrane teichoic acid goes up to the cytoplasmic membrane. Membrane teichoic acid is also known as lipoteichoic acid. Coming to the composition of gram-negative cell wall, we have seen that peptidoglycan layer is thin, that is 2 to 3 nanometers. Outer membrane is a bilayered structure and it has various proteins, especially the porin protein, which helps in diffusion of small molecules. Then it has a lipopolysaccharide. We have already covered that it consists of lipidae, polysaccharide core and O antigen, and finally the lipoprotein. Now what is the function of this cell wall? This cell wall provides shape, size, rigidity to the bacterial structure. It is helpful in cell division. There are various antigens on the surface of cell wall because of which there is virulence, and it provides immunity since antibodies are formed against these antigens. And then it also provides endotoxicity, which is a function of lipopolysaccharide. Endotoxicity means various functions like lethality. Now we have come to the MCQ portion. So the question first is, the region of bacterial cell where circular chromosome is packed is known as A. Nucleus, B. Nucleoli, C. Nucleoid, or D. Nucleoplasm. So the correct answer is C. Nucleoid. Coming to the next. Gram-positive bacterial cell wall is thicker than gram-negative cell wall, is a multi-layered structure, it has teichoic acids, or all of the above are true. So the answer is all of them are true. We have seen that gram-positive cell wall is thicker as compared to gram-negative cell wall. Which of these structures is present on cell surface of gram-negative bacteria? A. Lipoteichoic acid, B. O antigen of lipopolysaccharide, C. Electron transport system proteins, D. Peptidoglycan layer. Which of them is present on the surface? Answer is B. O antigen of lipopolysaccharide. We have seen there is a lipopolysaccharide which consists of lipidae, polysaccharide core and O antigen which is present on the surface of gram-negative bacteria. Fourth question is, lipoprotein in gram-negative cell wall connects outer membrane with inner membrane. It connects outer membrane with peptidoglycan layer, connects the outer membrane with teichoic acid, or connects outer membrane with porin proteins. The correct answer is B. It connects outer membrane with peptidoglycan. Teichoic acid is not present in gram-negative, so this option is totally wrong. Question number 5. Which of these is not a function of cell wall? Cell wall provides rigidity, it has antigenic determinants, it helps in protein synthesis, and it is involved in cell division. Correct answer is C. Protein synthesis. Protein synthesis is not a function of cell wall, it is a function of ribosomes, which we will be discussing later on. So the last question is, which of these do not have a cell wall? L form, mycoplasma, protoplasm, D. All of the above. And the correct answer is D. All of the above. As we have already seen that L form, mycoplasma, protoplasm and spheroplast, all of them have no cell wall. They are cell wall deficient or defective form. Thank you. If you like the video, kindly hit the like button and subscribe to the channel.